those will become a individual organisms, which we have some of them in here. That's pretty much what I'm referring to here. Okay, I don't know if Dr. McRae talked about it or not, but anyhow, that's the story behind identical twin. Metamorphosis, sharp changes in embryonic development. It means in case of arthropods, we humans do not undergo metamorphosis because when you have an embryo in a mother's womb, it is you have one head, two arms, two legs. So, but in other cases, in arthropods, they go under sharp changes from egg, then you have the larva, then you have the pupa, then you have adult. Because you can see the changes are drastically. This does not look like adult. This does not look like adult. That's what they call this metamorphosis. And I hope as we age, our brain undergo metamorphosis. I look at my mentality when I was like 20 years old. It's complete. I'm thinking about complete different things than when I'm right now 50 years old. So I believe, I think, I don't know, some people say my mind, my thoughts went under metamorphosis. Okay, great. This is the diagram I would like you to know. I summarize it. When you take, later on in your life, when you go to a major school, Davis, UOP, I don't know, wherever you go, Riverside, UCLA, I had a former student who went to UCLA, he just came by, they visited. And then you take a course by name of embryology, you guys. They talk about this all semester or all quarter. I had one student who went to medical school in Nebraska. He said, Amir, thanks to your half-an-hour lecture or 1 hour lecture on embryology, I'm one step ahead of my classmates. Just imagine, the whole entire semester, they're going to talk about embryology. But just this will get you ahead of everybody in your class. So I hope you know what I'm talking about. So you have a blastula right here, okay? Right here, sorry, I don't know if this is blastula. Right here, so you have a blastula. Blastula becomes a gastrula. In case of human, I'm referring to human. And then gastrula undergo different germ layers. If I may, here is a tadpole. Here is the tadpole. On the models we have in the lab, anything that is yellow is endoderm. Anything that is blue or brown is ectoderm. Anything that is red or orange is mesoderm. In the lab, anything that we have that is brown or blue is ectoderm. Anything we have yellow is endoderm. Anything we have red or orange is mesoderm. All of the models in the lab. So, what happens after gastrula? Remember, this is a gastrula of the starfish. And then we have, let me show you this is a frog model of frog. Right here is a zygote. Look at the thickness of the yolk. This is the vegetal end. This is the animal end. Okay, so becomes two cells. Nobody took this one because I guess from far distance you cannot see this. This is two balls or not. They want to hang it up from the truck or something. Don't know. 4 cells. This is all morula, morula, morula. And look, you see that opening right here? So what stage is that? Blastula, definitely. Because that space, that empty space, empty full of fluid, is blastocele. Remember? Yes?
So the white part on that model is the yolk?
Yeah. Okay. Yeah. Still, it undergoes, it's not, at this stage, you see some cells in here. They call it a vegetal pole, the side. North pole, south pole, vegetal pole. Okay? Still, at the beginning, it does not go under mitosis cleavage. Okay? So you still have blastocele, right? Blastula, blastula. The cells are becoming smaller, but the size stays same. But now look at this. I see different stages. I see gastrula. In the gastrula, This is gastrula. Why? How do I know? Because I have mesoderm, endoderm, ectoderm. Okay? So the yellow is endoderm, the red, orange is mesoderm, and the brown is ectoderm, and so on and so forth. So this is gastrula. Of course, the next stage is also gastrula, and then the gastrula moves on and on and on. So what does the ectoderm becomes? Ectoderm, remember in this model, blue, this is a bigger version of the tadpole. The blue is ectoderm. The blue becomes your epidermis, associated structures of the hair, nails, the brain, and nervous system. So you should know that. And then the endoderm, the yellow, in this model, the yellow becomes your inner lining of the GI tract, gastrointestinal tract, GI, inner lining of respiratory tract, you all know respiratory tract, and glands including liver and pancreas. They all come from endoderm. And then mesoderm is a whole bunch of them. The dermis layer. You remember the dermis layer, right? For the first exam material, I wrote it down in my notes in the PowerPoint. Dermis comes from mesoderm, and epidermis comes from ectoderm. I said, don't worry about it. We'll talk about it next exam material, right here. Today's the day I'm talking about. Okay, so, and then mesoderm, you have somites, which you will see it in the lab. There are repeated segments in the animal, like your ribs. It would be muscles, it would be your somites, become muscles, outer covering of inner organ, excretory system, which is your urinary system. And then mesenchyme, loose migratory cells, become dermis, of course, your bone, cartilage, circulatory system, including heart, blood vessels, etc. They all come from mesoderm. Okay, so those are the structures on the quiz on Thursday. I might ask, heart comes from which, or matching, ectoderm, endoderm, mesoderm. Yes?
Is that like the chronological order they
No,
are?
occurs at the same time.
Okay.
When you have, it's not like you're not,
Yeah, like I
you're
didn't
asking
like
a good question. It's not morula, blastula, gastrula. When gastrula occurs, then you have the 3 germ layers. When gastrula occurs, I put it away, no, I did not, right here. When gastrula occurs, then you should have the 3 germ layers.
Ectoderm, mesoderm, and endoderm occur at the same time.
Right. As the embryo is developing, becoming bigger and bigger and bigger, then they are occurring at the same time. But some parts, they appear later. I don't want to get into too much embryology. Some parts appear earlier in the development. Okay, but you have all 3 in the gastrula stage. You see in the gastrula you have all 3 of them. Okay. Then this is the development of the brain. If I run out of questions, because in the chick embryo, you have these terminologies. You have telencephalon, diencephalon, mesencephalon, metencephalon, myelencephalon, rhombencephalon. You have these terminologies. I thought, might as well, I put these, this PowerPoint so you can relate to it what the heck they are. Okay? So when you have in embryo, you have the prosencephalon becomes the prosencephalon. You will see it in chick slides. Become telencephalon and diencephalon. Telencephalon becomes your basal ganglion, olfactory sense of smell, and cerebrum. The biggest portion of your brain is your cerebrum. You remember that? And then your diencephalon becomes your pineal gland, biological clock. I've talked about it a little bit. Thalamus and hypothalamus. So you can relate better to these terminologies that I'm throwing at you in the lab. So what they become as the embryo grows, eventually. Am I making some sense, everybody? So these terminologies, you see them in the lab. So if I run out of question to make, I might ask, what does diencephalon becomes? Which of the following structures becomes diencephalon? You should know that. It will eventually help you in medical school because you have to know all of this. Of course, in more detail. Amniotes and amniotic eggs, reptiles, birds, and mammals, chorioallantoic membrane, amnion, allantois, chorion. Might as well, let's talk about it here. There are 3 layers, chorioallantoic membrane. These two, what am I saying? Later on during embryonic development, these two become one, which is called chorioallantoic membrane. What happens, you have amnion, look at it, this fluid outside of the embryo which protects the embryo. All of these 3 are to protect. And of course you have the allantois right here for gas exchange, food and nutrient exchange. That's what the function of allantois is. But the other, the chorion and amnion, it is for shock absorbance. What happened when animals quickly, a little bit of evolution, when animals moved from sea, of course there was an egg in the sea, it was surrounded by water. The water around it would take the pressure. If there was somebody beat that egg, you know, if the egg would flew away, or there was not much of a heart. But when animals came to the land, then the egg had to come up with a system not to be desiccated, number 1. And number 2, to protect the egg from a little bit of a shock. I hope I'm making some sense. Of course, you can take your egg and crash it. Yeah. But if you kick it a little bit, it does not break. Okay? When a pregnant woman, when mammals evolve, you still have that fluid in there. Okay? So the fluid is in there, somebody accidentally pushes so the baby is not aborted. I hope I'm making some sense. So reptiles, birds, and mammals, they moved to the land, and they had to come up with a system to protect the embryo. And what are the system? Amnion, allantois, and chorion. Okay, of course, allantois for exchange of nutrient. The eggs, the fish eggs in the sea, they have yolk. They have nutrition. Of course, these guys have yolk too. But in case of mammals, you have to be connected to the mother. So this would be your placenta and being connected. Again, I don't want to go too much into details, just a little bit of story. Formation of coelom, schizocilus, I already talked about that. Advanced chordates from schizocilus. Is that right? We talked about that. Is there any question on that one slide? I talked about all of them. Okay, a little here, let's talk about, this is a joke, so I presented this joke to some conferences, some meeting, and some people got offended. Oh, you're being male chauvinist. No, I'm not being male. You're being female chauvinist. No, I'm not being female chauvinist. You're being the joke. A little girl grunts this, dad, say, Dad, where did I get my intelligence from? The dad look at the girl. The father replied, well, you must have got it from your mother, because I still have all of mine. Half of it is from your father, half of it is from your mother. But let's go talk about the next 2 concepts that I would like to talk about. It is the Holly, Dolly, not Holly, sorry about that. Dolly the sheep, I don't know, you've heard the story, they cloned, for you probably were not born then, 1997, 98, so 1997. Yeah, some of you were not born then. What happened, it was a breakthrough in science. Before that was, before this study, the breakthrough, it was the PCR. That was when you can multiply DNA by a machine. That was a breakthrough. Nowadays they still use it. Before PCR, if you wanted to multiply a DNA, you had to use bacteria. I'm not going to get into that. There was a lot of things that could go wrong. But anyhow, this happened, and they worked a lot on it. And what is the idea of cloning, you can clone animals, mammals. Before that, they cloned other animals like frogs, other simpler organisms, but this was the first mammal they could clone, a sheep. So what happened, they did this. They took the egg, one egg from a black-headed sheep to make sure this is happening, and they took the nucleus out. So you had the egg, which had all of the machinery for becoming an adult organism. Am I making some sense? They took the nucleus out, and then they took another white-headed sheep. They took the cells from mammary gland, and the mammary gland has, Nicholas was talking about ploidy, has all 23, did I mention in here how many chromosomes?
54.
54 chromosomes divided by 2 is what? And all, did I mention it? I guess not.
26.
It's in my other PowerPoint.
27.
I have it. But anyhow, it had all of the pairs, let's say 23, same as human, had all of 23 pairs of chromosomes, okay? So they took the nucleus out of the mammary cells. Nowadays they do it differently because of technology has improved, they played with it, they can do a whole bunch of stuff. So they took the nucleus out of the mammary cells and they infused it with a syringe into the empty egg. Do I make sense, everybody? Or should I turn off some lights? I'm afraid I turn off some lights. You had lunch already and you fall asleep. Just like back in 19 something. I don't even remember. So when they put the egg in there, when they put the nucleus in the empty egg, then they give it a shock, electric shock. Because there is a theory when in fallopian tube of females, sperm reaches the egg and wants to enter the egg. After it reaches the egg, there is a shock. They are not sure what kind of shock that is, but since they knew that and they are working on it, they gave it the electric shock. Maybe the cell wakes up. So they gave it the electric shock. Luckily, it went from 1 cell, right? They did it in laboratory, to 2 cells, to 4 cells, to 8 cells, to a nice blastula, and they took this blastula and put it in a sheep, any sheep. Gestation period of sheep is, let's say, 6 weeks, 7 weeks. I'm taking up, it's up there. So the cell can come. After 6, 7 weeks, this sheep was born. And guess what? This sheep was exactly identical. I'll go over that in a minute, hang on. 95% identical to which sheep? Black-headed sheep or white-headed sheep?
Yes.
White-headed sheep. White-headed sheep. Because these chromosomes, is that right, came from those 23 pairs of chromosomes came from that sheep. So they used the eggs of that sheep. I hope I'm making some sense. And they came up with the exact clone, 95% clone of this one. Why I say 95%? What is the other 5%? Very good, mitochondria. And nowadays they're working on that. They infuse some mitochondria. This is they were not able to take the mitochondria. Nowadays they do it differently. They take the nucleus out and they put the whole entire cell into the egg cell. So their whole days, and instead of electric shock, they're giving it a acid or base shock. They're playing around with it a lot differently nowadays.
So if they use an egg and so if they use an egg from the same pit animal they got the chromosomes from the mitochondrials were the same.
Sure, you can do that. So the women in this room, they can clone themselves. Exactly. Same. We can. We men cannot do that.
Wasn't the
we can clone ourselves, but 95%, because their mitochondria is going to be in the egg.
Wasn't Dolly born with some genetic defects?
Yeah, Dolly died. Well, usually what happens, okay, since you mentioned that, Dolly died, and usually the life period of sheep, normal lifespan of sheep is 19, 20, 21. Dolly died after 5 years because right now all of the cloning they do the animal has to be, gives birth by C-section, because for some reason or other the clone animal are bigger than what they are supposed to be. And then Dolly died because they used chromosomes of a, let's say, 10 years old. Imagine, the lifespan is 20 years old. They use a 10-year-old sheep. So telomeres has lost their, you all know what I'm talking about.
Yeah.
Telomeres has lost. So that's why Dolly had so much problem. That's why all of clone animals have so much problem. They usually die early on because of two things that suggest in C-section, number one, not every C. A lot of us are here born due to C-section, but none of us are chromosomes. Was a 20, 40, 50 years old man or woman chromosomes. Okay. So, but that's the C-section. They're saying maybe has something to do with it. But the chromosomes, it's an older chromosome. They have some problems which they don't know why the animal is dying. Okay, so quickly, that was cloning. And totipotent is one cell can become the entire organism. That's the definition of a totipotent. When you go to totipotent, it would be equivalent to a freshman in college. When you're a freshman in college, if you don't know your major, you start taking classes and you can go any major you want. That's what the egg is. The entire egg, if it is manipulated, it can become entire organs. Am I making some sense? That's what the term totipotent, the total, toti came from total, potent, potential, potency. Oh, we have we have stem cells in ourselves, in our bone marrow. We've already talked about that. So the multipotent potential stem cells in our bone marrow can become B cells, monocytes. You already know that. Okay, so, and eventually some of them become white blood cells, red blood cells lose the nucleus, right here. They lose the nucleus and they become red blood cells. So your red blood cells at one time had nucleus. After a period of time, your red blood cells, you lose the nucleus. Multipotent or pluripotent cells, stem cells that can drive to multiple cell types. And what they do in a laboratory, what they do in laboratory, there are two types of stem cells. One of them is embryonic stem cell. Here's a blastula, and they take the cells here. These cells will become placenta, so they're no good. But they take these cells and put them in a dish, a petri dish, and they add solution to it, and then eventually they can get liver cells, neuron, or heart muscle cells. Any cells you want, any cells, okay? It depends on what you add to the nutrition of that cell. The other one is they take the cells from any part of your body, your fat cells. In this case, the diagram they're showing you, they take it from bone marrow. They can take liver cells, almost every type of cells they can take from your body, and they add nutrition, culture it, and then it can become the cardiac cells, neurons, hepatocytes, which you already know about that. Which one you think it's better? Huh? Embryonic cell, stem cell, yeah, it's better, but embryonic stem cell, they do not have much challenge, scientists at the present time, they do not have much challenge to make a liver cell out of it, to make a neuron out of it, to make a cardiac. It's easier because the cells have not been determined. Am I making some sense? But if we do that, and I need heart cells, can they put these cells into me, into my body? Why not?
Immune rejection.
Yeah, my immune system will reject it. So it is better they take my fat cells or my bone marrow cells and go this route. Do that make sense? Then I will not reject it because same cells. I hope I'm making some sense. So having embryonic stem cell has its own disadvantages. You can easily grow heart cells and give it to the patient who needs heart cells. okay? But they're going to reject it. They have to give them a set of drugs to counter fight the rejection. I hope I'm making some sense. So stem cells, I mentioned only two. There are different type of stem cells. I mentioned only here two type of stem cells, embryonic stem cells and adult stem cells. So make sure when you study your questions at the beginning, you can answer everything here. Okay, this is the 33 hours chick embryo. I'm not going to go over the parts. These are somites. This is heart, somites here. Make sure you study them in the lab and be ready. This is the entire organism, the 33 hours chick. So what happened? This is the entire animal. On your slides, you will see something like, say, it says whole mount, WH. Whole mount, sorry. Whole mount, it means the entire animal. Cross section, it means they cut the animal in the cross and they put it in there. Longitudinal section, they cut the animal longitudinally and put it on the slide. And you have serial section. Serial section, it means they cut the animal in series. They cut it through the head, they cut it through the heart, they cut it through the somites, and they cut it through the posterior end of the animal. This is a serial section. You have this slide. And then you have the whole mount slide of 33 hours chick. 33 hours chick. They go back there. Right here. This is the whole mount, the whole entire chick. After 33 hours, you're putting in an incubator. This is what comes up, 33 hours. And then this is the serial section. They cut it through the head, through the heart, through the somites, and then through the posterior end of the animal. Okay? And that's all you have to know. All of the terminologies in there, I would like you to know it for lab practical quiz on Thursday. Okay, then the next one, let's move on and get out of embryology a little bit today. The next one, Robert Whittaker. I threw that term at you guys. Robert Whittaker proposed back in 1960s. This is what he proposed: that prokaryotic cell, right here, bacteria, evolved to cells that have nucleus. So this is still true. Most scientists believe that the very first cells that appeared on the surface of the earth, they were organisms that they did not have nucleus, okay, such as bacteria. And then these organisms, they evolved to single-cell organisms, which we still have them on Earth, on planet Earth, and they are single-cell, and they have a nucleus, okay? Then what happened, he proposed that the organisms that are protista, they have nucleus, some of them evolved to become animals. Some of them evolved to become plants. Some of these protista evolved to become fungi. It wasn't like animal became plants, plants became fungi, or the other way around. Fungi became plants, plants became animals. None of that. The protista, okay, the single-cell organisms, not all of them are like that. Ball box, you're looking at it in the lab. It's a colony of cells. But anyhow, they evolved to plants, some of them evolved to fungi, some of them evolved to animals. Classification of animal kingdom. Quickly, here is Coelom. There are about 1.5 million species. That is not true anymore. It's about 1.92 million species that have been discovered, named. I'm not talking about viruses. I'm talking about just bacteria, all the way from bacteria to animals in Africa. About 1.5 million has been discovered, and there is a lot more. About 2 million have been discovered. First person who came said, hey, let's give it a shape. Let's be able that you and I can communicate it. It was Coelom. He was a medical doctor. He said, let's give all of the living organisms on planet Earth a binomial nomenclature. Give it two names, genus, species. based on comparative anatomy. It means he compared structure of the organisms and he put them in the groups that they are supposed to be. And then the comparative anatomy comprised of kingdom, phylum, class, order, family, genus, and species. So you should know that order. I would like you to know the order because the rest of semester is going to come to our rescue. So kingdom, we have animal kingdom, plant kingdom, fungi kingdom, and then phylum. In that kingdom, there are a few phylum, and the phyla is plural. And in that phylum, there are classes, there are order, and the order, there is family, genus, species. It's just like a continent. You have a continent. Inside of the continent, you have countries. Inside of countries, you have states or province, whatever you have. Inside of the province or state, you have cities. In the cities, you have streets. And in the streets, you have blocks and the neighborhood. And then that would be your house. I'm making an analogy. They make this classification, they make this taxonomy so we can communicate better. That's what Carlos Linnaeus was after. Okay? And then the way you write scientific name, you guys, the first word is the name of genus, right here, and genera is plural. It's written in capital initial letter. The second word is small letter, all small cases. Both words are italic or underlined. So during lab practical exam or lecture exam, you must underline. You cannot say, Amir, look at my handwriting, this is italicized. I will not take it. You must underline it, the scientific names. So what do you underline? Only genus and species. Again, I thought I fixed my notes. There is no line here. There is a space, Homo sapiens. Homo is the name of the genus for human. Sapien is the name of species for human. But here we go, it's a space here, gorilla. I thought I got rid of them, I fixed them, but I guess not in this PowerPoint. Okay, sometimes in some species you see 3 names. The last one is called a subspecies because the Trypanosoma brucei, in these, I will talk about them later on, but they are very close to each other. Only by DNA, by looking at them a little bit under the microscope, you'll be able to, or where they came from, you might be able to see them differently. So sometimes, instead of somebody wants to write down Trypanosoma brucei, or then CSA Trypanosoma brucei, Gambiense Trypanosoma brucei, they just write down Trypanosoma SP, period. It means all of these 3. Okay? Gorilla can also be written after you write the first time. Spell it out. The second time you can write down G dot gorilla. And you must still, second time, underline it. Okay? Trichomonas vaginalis, same thing. Underline or italicize. Phylogeny is based on homology, not analogy. Phylogeny means classification of organisms. It's based on how similar they are, not on how they function. For example, you don't put a butterfly and a bat in the same group. They both fly, but no, they do not go in the same group. Butterfly goes in insects and bat goes in vertebrates. Okay, I hope I'm making some sense. Human, of course, Robert Whittaker. This is the classification of human. I would like you to know that. Human belongs to phyla. They are in animal kingdom, for example, just an example, 33 phyla. Human belongs to phylum Chordata. Tetrapoda is a subphylum. Class Mammalia. Human belongs to subclass Thera. And then order Primates. And family Homondiae. Genus Homo. And species Sapiens. So I would like you to know that. I might grab your collar in a mall or movie theater and say, what is the name of the order for human? And you might say, Amir, you're out of order. If I grab your collar and say, what is the name of the human for human in a movie theater, I say, oh, Amir, you're out of order. Call cops. So or human belongs to primates. The name of the order for humans. These are animal kingdom. Okay, let's stop. So questions at the beginning. I went all the way to diagram, gem layers, brain, purpose of amnion. Oh boy, these are all easy. Covus Linnaeus, order, taxonomy, rules, Robert Whittaker. Oh boy, I answered all the way to question number 22. 22 I did not answer. Now somebody's going to, I mean, is 22 going to be in the quiz? No, 22 is not in the quiz. 21 is in the quiz. And I already said the slides at the beginning. Is there any question before we